مرحبا جميعا وأهلا وسهلا فيكم معنا اليوم. قبل ما نبدأ بالسيشن حابة أعرفكم بشكل سريع عن جمعيتنا JPSA. JPSA هي جمعية غير ربحية متابعة لأي جهة سياسية أو دينية، تأسست بالأردن عام 2007 وهي تابعة للاتحاد العالمي لطلاب الصيدلة IPSF. بتضم بالأردن 4 جامعات: الجامعة الأردنية، جامعة العلوم والتكنولوجيا، الجامعة الهاشمية، وجامعة الشرق الأوسط. أهداف الجمعية إنها تزيد انخراط طلاب الصيدلة بسوق العمل وتزيد التواصل ما بين طلاب الصيدلة من مختلف الجامعات، وتعطي طلاب الصيدلة الفرصة لحتى يشاركوا ب different national و international events، و it builds partnerships with different leading institutions. هذا البورد للجمعية لسنة 2020-2021. Our portfolios: عندنا فريق الصحة العامة اللي بيعمل حملات توعية صحية عن مختلف الأمراض المنتشرة بالمجتمع. عندنا فريق الحملات الإنسانية اللي بيعمل حملات تطوعية تخدم فئات مختلفة بالمجتمع مثل كبار السن والأيتام. عندنا فريق السوشيال اللي بيعمل أنشطة ترفيهية لطلاب الصيدلة وبيساعدهم إنهم يتعرفوا على بعض أكثر. عندنا السب اللي هو الستودنتس اكسشينج بروجرام، بيعطي طلاب الصيدلة الفرصة حتى يتدربوا برا الأردن، وبيعطي الطلاب اللي برا الفرصة إنهم يجوا يتدربوا عنا بالأردن. واخر شيء عندنا فريق البروفيشنال ديفلوبمنت اللي بيعمل على تطوير مهارات طالب الصيدله بمختلف المجالات، وكان له دور كبير بتنظيم المؤتمر لهاي السنه. هلا عندنا سيشن الإبيجينيتكس مع الدكتوره عايشه الرفوح. هي PhD candidate بيونيفرسيتي اوف ايوا، وهي بتشتغل ريسيرش على موضوع الإبيجينيتكس. دكتوره بتقدري انك تبلشي معنا هلا وتعرضي السلايدز؟
Sure. Okay, hello everyone. Can you see my slide? Hello?
Yes.
Okay, cool. Yes. Hello everyone. I am very happy actually to be part of this and participate with you and attend this meeting and have my talk. And I'm also graduated from University of Jordan, so I'm so proud of my colleagues there at the University of Jordan College of Pharmacy. They're like for arranging these meetings in professional way. Yesterday I was talking with them and asking them, are you graduated, are you working? And then it's like, all of us are students. And I was like, that's amazing how like you arrange all this and contacting people and I like everything about that. So thank you guys, and I'm so happy to have my talk as one of the graduated of the University of Jordan College of Pharmacy, and also as a PhD from the College of Pharmacy, University of Iowa. Today I would like, I'm not going to talk about one of my research project. I would like to give more general talk about the DNA methylation as a one of the epigenetics mechanism. So let's get started. So today this is like the topics. These are the topics of today's discussion. We have DNA methylation as a biomarker, DNA methylation as a drug target. And we'll start with these 4 questions about DNA methylation. So what is DNA methylation? What are the enzymes responsible for DNA methylation synthesis? Where can DNA methylation occurs, and what's the function of DNA methylations? So we'll start the first thing with the epigenetics. So what is epigenetics? Epi mean above. So when I say epigenetics, there are certain heritable changes that happen in the gene function without changing the DNA sequence itself. So this is how like the chromosome. Here's the chromatin. The chromatin is wrapped around the histone protein, and like this, like the double helix DNA. Okay, and this the nucleotide. When you know, like thymine is with the adenine, guanine with the cytosine, and here's like just the basic structures of our genome. So when I say EB, that's mean above. So there are certain heritable changes that happen above the DNA sequence without changing the DNA sequence itself. So wherever the DNA, it's like the arrangement in the double helix, C next to C or T or like whatever the arrangement, there is no change in the DNA sequence itself. It's happened above the DNA sequence. That's what's called epigenetic mechanism. So these, the most common example of the epigenetic mechanism are DNA methylations and histone modifications. These are the histone. They are, a protein wrapped, like the chromatin are wrapped around these proteins. And if there is some kind of a changes to the protein, can affect the genome. Okay. The same thing with the DNA methylation. So DNA methylations is like something happened above the DNA sequence, as shown in this figure. These, like the red dot, if you can see them, these are the methylation. We're going to talk more in details about this. For the purpose of our talk today, we will focus on DNA methylation because that's where my research focus more mainly in DNA methylation. So let's start with the DNA methylation definitions. So DNA methylation is a covalent transfer of the methyl group to the cytosine nucleotide by a group of the enzyme called DNA methyltransferase enzyme, DNMT. Okay, and usually DNA methylation happen when the cytosine next to the guanine in the linear nucleotide sequence. So that is called CBG site. Why we are calling it CBG site, not CG? Because if I'm saying CG, that means I am talking about that C is complementary to the G. I'm talking about the bear. But when I say CBG, I'm talking about that C and the cytosine and then a guanine nucleotide are in the same strand. They are next to each other. So when the cytosine followed by guanine, that's called CBG site. And we use the P as like to distinguish between the bear when I say like CG as shown here. Okay. So DNA methylations happen usually when the C next to the guanine in the same strand. As I mentioned, DNA methylation happens by the help of the group of the enzyme called DNA methyltransferase enzyme, DNMT. DNMT, there is a several isoform for the DNA methyltransferase enzyme. There are DNA methyltransferase enzyme 1. This enzyme is responsible for maintaining the existing methylation patterns after DNA replication. Let's look at this figure. Okay, so this our DNA. We have this like two CBG site. One of them unmethylated, the one in the right, the left one is methylated. We do want like now to do the DNA replication. So we want to have more copies of our DNA. Okay. After the DNA replication, there is a parent strand, which is the top strand, red strand, and there is the daughter strand, which is in the orange color here, the bottom strand. So after the DNA replication, one of the strand will have the methylations, but the other strand, because it's a new one, there is no methylation. Because in the synthesis, there is no, enzyme responsible for attaching this. Because the replications enzyme, they don't have the ability to add the methylation group to the CBG site. So should be another group of the enzyme called DNA methyltransferase enzyme 1 that's responsible for adding the methylation to the CBG site. And this is the nice thing about this, how this enzyme, recognize, we should add methylation here to the daughter strand, not the parent strand. They have sensitivity to the hemimethylated state, which is like this, whereas one strand is methylated, hemi is like kind of, you know. So, and one of them is methylated and the other one is unmethylated. by the help of the enzyme, DNA methyltransferase enzyme 1, we'll see like after like that, we're going to have the same pattern of the DNA methylations in the two strand, the old strand and the new strand. So our DNA now have the same DNA methylation patterns. And as you can see from the image, is like DNA methyltransferase enzyme 1 only add the methylation to the one already has a methylation group. So whatever I mean in the original DNA there is no methylation, it's still there is no methylation. There is also another isoform of DNA methyltransferase enzyme. There's 3A and 3B. These are responsible for establishing de novo DNA methylation. De novo DNA methylation, that's mean creating a new site of DNA methylations. So whereas like I mean, as shown in this figure, they have, I mean, like looking in this strand, there's like CG site, which is where usually DNA methylations happens. So after that, I mean, like DNA methyltransferase enzyme 3A and 3B will will create a new methylation site by transferring the methyl group to the cytosine. So creating a new site along the genome. So we can look at the DNA methyltransferase enzyme as like two categories. The photocopiers as a DNA methyltransferase enzyme one, and the creator. Like I mean the DNA methyltransferase 3A and the 3B. So I mentioned that DNA methylation usually happens with the cytosine next to the guanine, which is called in the same linear strand is called CBG site. There are like sorry, there's like where's how many, I mean in our genome there is like 28 million CBG dinucleotide, and 70 to 80% of all the CBG dinucleotide are heavily methylated. These CBG site actually in our genome they cluster in like in specific genomic regions, mainly in two regions. One of them is called the CBG island, the other one is the CBG shore. So look, I mean, at this graph, whereas like I mean the um, here I mean the black one, I mean bands is like the dot is a methylated CBG site. The other one is unmethylated CBG site. And as I said, CBG site it can cluster in the genome in specific regions. So in CBG island is actually there are a short stretch of CBG rich sequence, usually around 1 KB size, like 1000 base pair in size, and the uh, the shores there are regions also have a high density of CBG site. However, they are lower density in comparison to the CBG island, and they usually located um, 2000 base pair from the CBG island. The CBG island, as I said, is short stretch, 1000 base pair. 50 of these CBG island are located in the region of transcription start site. What is transcription start site? So we have our gene, we have so there's always like a point where it's like the gene starts transcriptions to create a DNA, to create RNA, and from the RNA to create amino protein. So CBG islands, 50% of the CBG island in our genome are located in the transcription start site. And 70% of the annotated gene promoters is upstream regions from the gene body are responsible for initiating the transcriptions are with the CBG island. So CBG island are usually located upstream from the gene, whereas like the transcription usually starts. So, and they are usually unmethylated when they are located in the promoter regions. Again, promoter regions are, is upstream region responsible for initiating. Usually they are associated with the CBG island. And when I say CBG island, high density of the CBG, usually the CBG island that's in the promoter region are usually unmethylated. Let's see why they are unmethylated. So this is, the gene body. As I said, here's the intron, the exon, and there is like the stop and the start of the gene. So this is the segment we have to transcribe to create mRNA, and then from the mRNA the protein. The promoter is upstream regions from the gene body, from the start point, and they are has a role in initiating the transcriptions. If the methylations happen in the promoter regions, that results in decreasing the transcriptions. When I say decreasing the transcriptions, that means we're not going to have mRNA enough, we're not going to have protein enough, so we're going to decrease the transcriptions. If the DNA methylations happen in the gene body, not in the promoter region, which is upstream regions, that usually results in increasing the transcriptions. So DNA methylation has a function in regulating the gene transcriptions. Let's take a closer look. It's exactly how the DNA methylations in the promoter regions change the transcriptions. So promoter, if there is a DNA methylation, so the blue one is the methylated CBG site. If they happen in the promoter regions, they will result in remodeling the chromatin. You remember the chromatin when I said it is a DNA that's wrapped around histone protein? So if the DNA methylations happen in the promoter regions, that will result in decreasing the transcriptions. Why? Because the chromatin will remodeling the chromatin in a closed state. So can you imagine, like, if you're looking here, it's difficult, like, to do transcriptions. Like, let's imagine, some, enzyme and transcription factors, they want to like bind to the DNA. Since they are in the closed state, the access of this is not an easy, like when it is in the open state as here. So unmethylated promoter regions will result in chromatin remodeling, changing the shape of the chromatin to have an open state, which is allowed to access of the transcription factors and the enzyme to transcribe the DNA and having mRNA and then later protein. Let's get a closer look to how DNA methylation inhibit the gene expression or transcriptions. So there is two ways, direct and indirect way. The top figure, it's the direct way. So here's the gene, here's the promoter regions, there's the methylated region, methylated promoters. The methylations at the promoter regions prevent the binding of the transcription factors. So the transcription factors, they are responsible for initiating the transcription. So we'll have more gene expression. If there is a DNA, excuse me, there is a DNA methylations at that regions that will result in inhibiting the transcription binding, and that result in inhibiting the gene expression. In the indirect way, there is a specific kind of protein called MBP. This protein is a methylation binding domain protein. So these proteins have the ability to bind to the methylated CBG site, but they are not. Like the transcription factor, they recruit another co-repressor complex, which is induce the closed state of the chromatin and inhibit the gene transcriptions. DNA methylations happen in our genome And there's a biological role of the DNA methylation in a specific biological process, such as chromosome inactivation, silencing of repetitive element, and genomic imprinting. I will not go in details in these. This is just like um, I'm just presenting this to be familiar with these terms that are associated with the DNA methylations. But there are like more details about this biological process, that including DNA methylation. So also DNA methylations change in the diseases. This is a group. I mean, this is just a list of some of the diseases with the symptoms and the etiology. So all these diseases actually they are associated with a change in the DNA methylations. So DNA methylation is not only as a biological process, is also associated with the many diseases. And for the purpose of our talk today, we'll focus in DNA methylation in cancer. However, I'm not doing DNA. I'm not studying DNA methylations in cancer. I'm studying it in my PhD for uh, psychiatric diseases. But as a one, DNA methylation is well established in cancer. So this is what our talk will talk. I mean, we'll focus in the DNA methylation as an example in cancer. So here is, I mean, like how DNA methylation play a roles in the cancer. Let's take a look. So here's, I mean, the normal cell and tumor cells. Usually in the tumor cells, there's like a hypomethylations in the repeated sequencing. When the repeated sequencing, that means like the same sequence is repeated. I mean, many times the segment of the DNA has like the same repeat. So usually in the tumor cell, the repetitive regions are hypomethylation. When I say hypomethylation, that means less methylations. They are unmethylated. Kind of like, it's not like, I mean, they fully methylated or have more methylations. They have less methylation in comparison to the normal cells. So here is the comparison between the normal cells and the tumor cells in two regions. Well, let's focus in two regions. CBG island, where they are usually, as I said, they are in the promoter region, which is upstream regions of the gene, and we'll talk also about the repeated sequences. So in tumor cancer, normal cells unmethylated the CBG site in the promoter region in the CBG island, because we want like to have more gene transcriptions. We want to have gene, I mean, to transcript this gene. In the tumor cells, there are actually hypermethylations, because, and that is of the tumor suppressor genes. So there is a kind of a group of the genes, it's called tumor suppressor genes. I mean, these genes are responsible for suppressing the tumorigenesis process in our body. If we do the hypermethylations, that means we're not creating a copies of this. So by not creating, that means we don't have a tumor suppressor as a protein that make our bodies that makes our bodies more susceptible to to the tumorigenesis process. So in the tumor cells, there's a hypermethylation in the promoter regions of the tumor suppressor gene, and hypomethylation in the repetitive regions or sequencing, which is different from the normal. We have a hypomethylation in the promoter regions, because we want these genes to transcribe to protect our body, and hypermethylation in repetitive regions, because we don't need this repetitive region to transcript it. Here's um, a summary of our first discussion, which is DNA methylations. DNA methylation can have, as I said, it's can have, can be defined in different in different ways, classical and biological, chemical and drug discovery. So we talk about the biological, it's like it's a regulation of the gene expression through the chromatin remodeling. It is, we talk about the chemical, that is reversible structural modification of the DNA and histone protein. And we talk about the classical, where we're saying alteration in the biological phenotype. It's changing the gene function without underlying the change in the genotype, without changing the DNA sequence itself. So for the rest of our talk, we'll focus in DNA methylation in drug discovery, because we'll talk about DNA methylation as a potential drug markers and a potential as a drug target. And the question now, the question now is, DNA methylation is a good potential biomarkers, and that is the next topic of our talk. Before starting answering this question, let's define what's biomarker, what's the ideal biomarker, and then get back to our question. So biomarkers is a molecular target analyzed in quantitative and qualitative manners that help us to detect and diagnose the response, the presence of the disease, and predict the outcome and the response to the specific treatment. So biomarker can help us and detect and diagnose the disease, or predict the outcome of the disease, or the response to the specific treatment. Biomarker can be classified in two categories, and that depends on if it's associated directly with the causal pathway or not. If it's associated, it's not associated, it's associated with the disease but not directly in the causal pathway. So that means is that there is a specific pathway that's associated with some disease, but we're getting the biomarker, like changing some proteins that not in the causal pathway. It's called descriptive biomarkers. The mechanistic biomarkers, where they are directly involved in the disease pathogenesis. And the ideal biomarker should be specific, sensitive, and detectable in specimen obtained through minimally invasive procedure. Because the thing is, like, we can have a biomarkers and ideal biomarkers, but they are like, we should get this from specific tissue, or like, as someone working in the psychiatric disease, the ideal is like to getting a tissue from the brain, but that's not visible, because uh i mean unless they are a post-mortem tissue. i mean so for that we need like to have um, to get our biomarkers from invasive procedure, definitely, and like detectable in a preferred tissue such as blood or urine, or another specimens such as urine. So these are the ideal biomarker criteria. So going back to our question, let's answer this question: is DNA methylation is a good um, uh, can be used as a biomarker, is a good potential biomarkers? And the answer is yes, for many reasons. The first reason, it is amplified and stable nature of the DNA. Because it's amplified DNA and the nature of the DNA, so we can have more copies of that. We can have enough copies to detect this in the blood. You know, Also, the methylation is chemically and biologically stable over time, which is something we want. It's like if you want to have a biomarker for specific disease, let's say like bipolar, and we know like there is a DNA methylation in specific genes, and that's associated with a bipolar disorder. So we're taking, blood samples. If the DNA methylation is not stable and is like after like 1 or 2 like days, the DNA methylation will, disappear from the DNA. Even if it is associated with disease, there is no way, like to detect that, because we have to take it in the specific time. One, that's not an ideal thing. With the DNA methylation, since it's a covalent transfer, so it is stable. Yeah, and it's chemically and biologically stable. So the other thing is the dynamic nature of the DNA methylation. What I mean, uh, the dynamic nature. DNA methylation is a certain heritable change epigenetic that happen above the DNA without changing the DNA sequence itself. So it's, and there is a group of the enzyme responsible for adding the methylation group to the cytosine. I mean, for many reasons, so like disease or biological process. So, I mean, there is a dynamic nature. Even we are not changing the DNA sequence itself, but the dynamic nature, we can have the DNA methylation there and not having it according to the circumstances of the body. If there is a disease or we exposed to some certain factors or like there's a biological process. So that is like that means like DNA methylation can differ with the disease status. So it can give us an indication of the disease. Another reason why DNA methylation is a good, biomarker because we can, detect the DNA methylation in a surrogate tissues such as a blood and other body fluid that can be obtained through minimally invasive procedure. So if we got the DNA from the blood or from the urine or whatever that body fluid or peripheral tissue, we still can see the DNA methylations. The second reason, why DNA methylation is good as a biomarker, it is the nature of the DNA methylation analysis technique and outcome. DNA methylation is usually when we measure it is a percentage. So it is not a binary, it's not yes or no. It is there is a range and it's a positive signals, but with a limited dynamic range. So we have a range from 0 to 1. It's not like we infinity. We have a range between 0 and 1. Still, we have a limited range, but at the same time, it's not binary. It's not like yes or no or a question. So it's kind of like, we can, for example, if there's like severity of disease or behavior or something, we can associate this with better with a limited dynamic range rather than having it's a binary yes or no. Also, DNA methylation independent of the total amount of the starting material. So we can detect the DNA methylation in the in the body fluid or in the peripheral tissue, independent of the total amount of the starting material. So even if we have, I mean, 10 nanograms of DNA or 50 nanograms, still we're going to detect the DNA methylation in the same way since it is a percentage. So there is no, not like we're having, oh, more DNA, we're going to have more DNA methylation. No, we're going to have, the same thing because we are the same value since we are measuring the DNA methylation in a percentage. No need for normalization to reference gene. And when I say that, I'm not quite sure, but if you are familiar with the RNA-seq, if you, which is like you're trying to measure the mRNA. So if we have the mRNA, usually they have to normalize it to the reference gene uh, to give an indication if there is a high expression or low expression. The last reason is there is several methods to analyze DNA methylations. And other reason, it's not the last one. There is also no requirement for any special handling. So if we got the DNA from blood or something, there is no special requirement for like, oh, it's fine. It's a normal process when you extract the DNA. It's the same thing, but the technique for studying DNA methylation is different. Also, DNA methylation analysis can also apply with a similar efficiency to fresh frozen and formalin fixed paraffin embedded tissue, and that's a great thing because, for for example, for RNA-seq, which is like sequencing of our mRNA to see if there is a gene expression or to test the gene expression of some genes. Usually it's hard to get this from the formalin fixed paraffin embedded tissue, but this is not the case with the DNA methylation. DNA methylation is perfect for this because we can get wherever like we got the DNA, it's from frozen samples or formalin verified fixed paraffin uh, embedded tissue, or even a post-mortem tissue, or like a DNA from the blood. Wherever we got the DNA, still we can detect the DNA methylation, which is great. Um, mention I like there's several techniques to study DNA methylation. I will not go in details about these techniques because I mean uh, there's many details about that. I mean mention these are like I mean the different technique, and this is how like I mean the steps for preparing the DNA for the studying the DNA methylations. I am using the bisulfate modifications. There is another methylation sensitive restriction enzyme, which is like, oh, the enzyme recognize where is the methylations and cut the DNA there. If there is no methylation, and no cut if there is methylations, and detect this later with another, I mean, technique, or the physical separation when we have, I mean, like the protein, the antibody that recognize the DNA methylation. So there are several techniques. I'm using the bisulfate modification sequencing for my studies, but as I said, there are many details about these procedures. So I will not go in details talking about these procedures. So for, as I said, as I mentioned, like at the beginning of the talk, I will focus in DNA methylation in cancer since it is well established there. So um, blood-based DNA methylation for cancer diagnosis is already, I mean, in the market. Cancer diagnosis identification of the cancer in either asymptomatic individual or patient with uncertain illness. I know some of you saying like, I mean, diagnosis of DNA for cancer is not that difficult. I mean, if there is a tumor, if there's a tumor, there's mean like there's, I mean, a solid tissue there, like you can that the physician can detect or like diagnose easily. But that's not the case, by the way, with all the cancers like types. There is like some cancer where like asymptomatic individual, and also there's like some kind of diseases that are have the like I mean similar symptoms as cancer, but they are not cancer. So how to distinguish between cancer, I mean, and non-cancer, I mean, diseases? So the question now, when do we need a laboratory-based biomarker for cancer diagnosis? We need when the symptoms are less well defined. Example, as I said, ovarian cancer has to be differentiated from benign lesion, cysts, and inflammation of the ovary, pelvis, and colon. Like all these has somehow similar symptoms. So to distinguish between these, we need, I mean, a biomarker. Here's like, I mean, the physician, I mean, by looking or, I mean, diagnose the patient is not enough to give, I mean, a differential diagnosis. We need, I mean, more accurate uh, based biomarkers, and DNA methylation can afford this. Um, Cancer screening, a typical procedure for diagnosis of the disease in a symptomatic individual because of multiple uncertainty regarding the nature of the disease. As I mentioned, like there's not always the time it's an easy to diagnose, like I mean the cancer, since like there's a symptomatic individual and there's another disease that have somehow similar symptoms to the cancer. So how we can use, I mean, blood um, DNA methylation for cancer diagnosis? Biological fluids, especially blood, containing molecular originated from in many different tissues of the body. So different molecules have been tested as systemic biomarkers. Um, and DNA methylations um, used as a biomarker for cancer diagnosis. And here's all the samples of some of the markets. These are actually the product for a DNA methylation based test for cancer diagnosis. and they are one of them, these two from the urine, the other ones are from the blood, and the diagnosis based on the DNA methylation changing for in the DNA. So now, I mean, we cover DNA as a methylation as a biomarker, and why we can use DNA methylation as a biomarker, and what makes, I mean, DNA methylation a good a good biomarker. The next question is DNA methylation as a drug target. As I mentioned earlier, you remember when I have these tables, when I have different immune diseases, symptoms, and things, and I said like these diseases are actually associated with the change in the DNA methylations. So if they are associated with the DNA methylation changes, can we somehow change the DNA methylations um, and like, I mean, help in, I mean, mitigating the symptoms of the disease, or, you know, so can we use DNA methylation as a drug target? So DNA methylation can be, I mean, as a drug target. That's not the thing I'm mainly working in my PhD. In my PhD, I'm mainly working in biomarkers. So I'm trying to see if there's a changes in the DNA methylation that are associated with the bipolar disease, schizophrenia, suicidal behavior. And I'm looking for these changes in the blood. And I'm trying to associate this with the disease to see if there's an association or not, and so aiming to have these changes in the DNA methylation as a biomarkers. But still, DNA methylation also can, I mean, be a drug target. Here is, you remember when I said like DNA methyltransferase enzyme, DNMT, they are a group of the enzyme responsible for transferring the methylation group from the cytosine, transfer the methyl group to the cytosine. So we have like methyl cytosine, the CBG site. Also, that's not the only enzyme uh, uh, that's not the only enzyme plays a role in the dynamic nature of the DNA. There is another group of the enzyme responsible for removing the DNA methylations and changing the DNA methylation status. And this enzyme, in addition to the DNA methyltransferase enzyme, can be used as therapeutic target. Other enzymes, the TET is responsible for removing the DNA methylations by having hemimethylations and going back to the cytosine without methylations. Another amino enzyme and mechanism. So maybe by targeting this enzyme, we can, I mean, change the DNA methylation status, and by this we can how like, I mean, be a drug target. DNA methyltransferase enzyme have emerged as attractive therapeutic targets. Several therapeutic agents that inhibit the DNA methyltransferase enzyme activity have already been identified, such as a cytidine. These drugs, I mean, inhibit the DNA methyltransferase enzyme. So instead of having more methylation, we're going to have less methylation. And this one of the drugs, I mean, compound that, I mean, have been identified as a therapeutic drug target. However, DNA methylation, we need more research to conduct, must be conducted regarding using DNA methylation as a biomarker and drug target with other diseases. That's only with the cancer. As I said in my PhD, I'm focusing in DNA methylation changes and with the psychiatric disease, including bipolar, schizophrenia, and suicidal behavior. And since it's how somehow it's established in the cancer, but still with other diseases, we need, I mean, more research to be conducted. And that is all I have for today. I would like to thank my advisor, Dr. Gain. And now I'm happy to answer any question you might have.
Thank you, Doctora. Does anybody have any questions? You can write your questions in the chat box.
Any question, guys? It's free today.
Thank you, Doctor. Mustafa says, thank you, Doctor. It was a very useful session.
Yeah, you're welcome, guys. I am, as I said, I am. Thank you for inviting me. I mean, it's so thanks for the invitations. It was great to see all this effort is from a student. They are still, I mean, struggling with the quizzes. I know. I have been there. I know. It's like each week's like at least 2 quizzes, so I know that. And you still have time to organize this and reach out to people all over the world. I really appreciate your work and effort. I am so proud of my colleagues there, and I'm happy also to participate in any future event if that's helpful for the student. And good luck with your organization and with your meeting. And I was so happy to be part of this. And have a good rest of your day, guys.
Thank you so much, Doctor, for being with us today. Thank you.
Yeah, definitely. Thanks, guys. See you.